Hello everyone. So this presentation is about bioinformatics. Our textbook defines bioinformatics as the study of data and information as it relates to knowledge within the context of the life sciences. Another definition found in our book defines bioinformatics as the field of science where biology, information technology, and computer science all merge to form one discipline. Because of its close link to biology, bioinformatics is said to also study the relationship between genotype, which is the genetic information that is associated with biological function, and phenotype, which is the observable characteristic, structure, function, and behavior of a living organism. Examples of phenotypes include eye color, hair color, height, and development of diseases. Bioinformatics uses computer science to create algorithms for promoting the development and testing of biological hypotheses. Examples of these include finding the genes of various organisms, predicting the structure or function of newly developed proteins, and developing protein models and examining evolutionary relationships. Some of the computational techniques used in bioinformatics include genomic sequence and molecular structural alignment, database design and data mining, macromolecular geometry, phylogenetic tree construction, prediction of protein structure and function, gene finding, and expression data clustering. Translational bioinformatics is the area in bioinformatics that is mainly focused on the use of bioinformatics approaches to address challenges in biomedicine and health. It focuses on the development of storage, analytic, and interpretive methods to optimize the transformation of increasingly voluminous biomedical data into proactive, predictive, preventive, and participatory health. So why is bioinformatics so important? Bioinformatics and bioinformatics techniques can help diagnose hereditary diseases, discover future drug targets, develop personalized drugs based on genetic profiles, and develop gene therapies to treat diseases with a strong genetic component. Because bioinformatics is so closely linked to biology and genomics, it is important to understand genome sequencing. To start, let's do a little review of biology. The human body is made up of trillions of cells. Humans have a pair of 23 chromosomes that carry genetic information. Chromosomes are found in the nucleus of each cell. Chromosomes are made up of double-twisted helices of DNA. DNA is composed of 4 sugar-based building blocks called nucleotides. These nucleotides are adenine, thymine, cytosine, and guanine, and are usually found in pairs. Genes are regions on chromosomes that encode instructions, which may result in proteins that then in turn enable biological functions. A complete set of genes is referred to as a genome. When scientists say that they have sequenced the human genome, they're basically saying that they have figured out the order of all the A's, C's, T's, and G's in sequence. The process of decoding genes involves transcribing DNA into RNA, as outlined in the first section of this example here, then translating RNA into amino acids that form the building blocks for proteins, outlined in the second half of the example here. Again, proteins enable biological functions. Okay, so perhaps the most well-known bioinformatics project is the Human Genome Project. The Human Genome Project was an international collaborative research program whose goal was the complete mapping and understanding of all the genes of human beings. The first draft of the human genome sequence was published in February of 2001, and the final sequence was completed and published in April of 2003. The Human Genome Project found that there are about 20,500 human genes. The Human Genome Project was so significant because it basically provided the world with a resource of detailed information about the structure, organization, and function of the complete set of human genes. This information can be thought of as a blueprint and contain instructions for building every human cell. So here is a list of some bioinformatics projects, databases, and centers. Some of these are private and some are governmental. I'm not going to go over each one because that would take up a lot of time, but these are all described briefly in our textbook if you want more information. So personal genomics. The decreasing cost of genome sequencing and the availability of personal genetics testing companies allow more personal uses of genetic data. Sequencing costs used to be $100 million per genome initially, and in 2017 it costs less than $1,000 per genome, according to the National Human Genome Research Institute. And the cost is expected to continue to decrease with advances in technology. The availability of personal genetics testing companies will give patients the ability to find out what their own genetic profile looks like, and gene testing can also be used to diagnose hereditary disease. Genomic information and electronic health records, or EHRs. In the future, a patient's genetic profile may become an additional data field in the patient's EHR. Genetic variations in a person's genome are what makes each person unique. However, recently, some gene variants have been found to be linked with disease, such as diabetes, Crohn's disease, rheumatoid arthritis, coronary artery disease, bipolar disorder, and many more. Clinical decision support tools integrated in the EHRs can help clinicians use genetic information to treat conditions with a strong genetic component. The October 2013 issue of Genetics in Medicine pointed out that for EHRs to support genetic information, they must store data in structured format, data must be standards-based, phenotypic information must also be stored as structured data, data must be available for use by rules engines, and EHRs must be able to display information needed by the clinician based on phenotypic and genotypic data. So it will be interesting to see if and how genetic information will be integrated within EHRs in the future. So here are some of the possible future trends in bioinformatics. First, new gene associations will continue to be reported because researchers are going to continue looking for correlations between genetic information and disease. Second, the cost to perform complete genome sequencing will continue to decrease, making it more cost-effective in the future. Third, the time to complete sequencing will continue to shorten. As a result, providers will have several ways to diagnose infectious diseases and birth defects. Fourth, companies will continue to offer more genetic testing each year and at a lower price. This may result in patient demand exceeding the ability to know what to do with the data. In the future, these companies will also be required to obtain FDA clearance for genetic testing. Fifth, there will be more decision support associated with both family history and genomic information that correlates with phenotypical information, risk factors, and lab results. And lastly, with the integration of genomic information within the EHR, there will be a better understanding of what turns genes on or off. Okay, so we finally reached the end of this presentation. Here's a list of the references that I used. Most of the information is found in our textbook, so you can refer to it if you have any questions. And here's just a list of the links to the images that I used. Well, I hope you guys enjoyed this presentation, and I hope it helped you gain a better understanding of bioinformatics.